A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the adaptive immune receptor repertoire community, also called the AIRR community. Our speakers today, Milena Pavlovic and Maria Chernigovskaya, are affiliated with the University of Oslo, as well as the AIRR community. Today, they will tell us about machine learning for the analysis of adaptive immune receptors and repertoires. Please note, the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our first speaker.
Hi, thank you so much for having us today. I'd like to start with a quote of a Ray Bradbury, but a bit modified. So, is the machine learning for the analysis of adaptive immune receptors and repertoires is just trying to find these things to see if they work, or maybe not? So today we will discuss different types of AIRR data and different machine learning algorithms that can be applied to this data. And also we will discuss the challenges and the best practices for machine learning in the adaptive immune receptor and repertoire data. As I said before, we will talk both about the receptors and repertoires. So immune receptors, or AIRRs, or BCRs, TCRs, they are one of the key agents of adaptive immune systems, and they can recognize different antigens. And one of the most important regions for the antigen recognition is called the CDR3 region, the most variable region. And adaptive immune repertoire is a set of different immune receptors within one individual. And it records all the past and present immune events that happening for an individual, for example, diseases, vaccinations, and other things. As we know, both BCRs and TCRs, they are constructed during the VDJ recombination process, where one V, D, and J gene is picked randomly to combine one chain of the receptor. And as we know, BCRs and TCRs, they are constructed of two chains. There are different types of AIRR data that we can use for the analysis. So on the receptor level, we can have receptor sequences. They can be nucleotide, amino acid sequences, full length, or just a CDR3 region with the name of the V gene and the name of the J gene. It can also be the three-dimensional structure of this receptor, or we can also know the binding of this specific receptor to a peptide. Most of the time, we know the sequence information for the receptor because the structural and binding information is much more harder to get. On the repertoire level, use AIRR sequences, pipelines. They can be bulk or single cell sequencing with or without UMIs. And sometimes we have pair data where for one chain, we know the other chain, which is coupled in one receptor, or sometimes we just have a set of unique heavy, light, alpha, beta receptors. There are many more interesting types of repertoire data that we can also use in the analysis. For example, we can couple paired AIRR-seq data with the gene expression information or with specificity to a set of predefined antigens. For example, Libra-seq analysis. But what are the main immunological questions that we want to address using those types of data? First of all, we want to improve our diagnostics using immune repertoires, for example. So if we have an individual and we know the repertoire of this individual, we want to know what are the diseases that this person has currently or had before. And on the receptor level, ideally, we want to improve our vaccines, monoclonal antibodies, and other therapeutics design, because we know that it takes a lot of time to develop a good drug, and potentially those data and those type of analysis can help us a lot. And there are many computational approaches that work with AIR data on the receptor level, on the repertoire level. Some of them are more simple, some of them are a bit more complex, so we can estimate the diversity of the repertoire, the architecture, or we can just predict the motif within the receptor. But all these computational approaches, they face the same problems, the same complexity of the AIR data. So first of all, we know that the potential diversity of an immune repertoire is extremely large, but when we have our sequencing data, it can only cover a tiny part of our potential diversity. Also, we know that the overlap between different individuals, between the repertoires of different individuals, is less than 1%. And on the receptor level, we know that one receptor can bind to several antigens and vice versa. And also similar receptors doesn't mean that they will bind to similar antigens, which complicates the binding prediction. So theoretically, all these problems can be addressed using machine learning, because we know that machine learning works well when we want to analyze complex high-dimensional data and detect some hidden patterns in those data. Now, Milena will talk about the machine learning a bit. Sorry, we have a tiny technical problem.
Yes, let's just switch. Okay, so when talking machine learning, we say this is yes. I hope you can hear me now. So when talking about machine learning, we first want to define what it is. What do we mean by this? So we can broadly define machine learning as a set of pattern recognition and function approximation techniques that find some sort of patterns in groups of typically large amounts of data. And we can also look at them as a set of different methods that allow us to make inferences about our data. So, for example, we could try to predict if the sequence will bind to a given antigen. And we can regard machine learning in this setting as a function approximation task. So we want to approximate the function that determines the binding from the sequence to the our target label. In this setting, we have sequences that are also called examples in machine learning terminology, and the function that we're interested in estimating and the target labels. In this case, sequences as such are typically not an input to a machine learning algorithm. So what we need to do is to represent our sequences or our data more generally in some format that could be used for machine learning. Some sort of numerical representation that carries the information that is relevant for our problem. So this representation or encoding could be manually set, like choosing different physicochemical features or some other type of information, or it can be estimated from the data. And also, when we have the function that the function that we want to estimate that performs our target task, we also have some parameters of that function, and they are typically learned during training. So one thing that's really important about machine learning is that it actually discovers statistical associations in the data. So these associations that exist in our features between that and our target labels are what enables good prediction. And of course, our aim is to get a good predictive model, also to get some sort of biological insight into how this function works. And this is why we also want our models not just to perform well, but to be interpretable. And it's important to note that what we learn here are not causal relations, so not really biological mechanisms, but they could be, this information can be a starting point for further analysis. And there are a lot of studies that employ machine learning for different types of analysis in the AIR data. And only, of course, a very small subset of them are shown here. So the next part that we want to discuss today is like, where exactly do we apply machine learning in AIR? And this is, of course, an exhaustive list of neither application areas nor the papers, but it's more aimed to provide an overview of what and some examples of what is happening in this field. So one example application area is predicting receptor specificity, the same as I showed before when defining machine learning. So one to examining this receptor specificity is to look into sequence similarity of particular receptors. So we can try to see which sequences are similar to the sequences of known binding, and use that information to predict the binding for new sequences. And there are many methods that are based on this information. For example, there was a TCR-DIST method in 2017, and there's a new version published recently where they look into how sequences differ and in which parts of the sequence they differ in order to compute the distance metrics between the distance between the receptors, and then classify the sequences according to their binding. Similar thought is kind of also behind the Glyph paper, again from 2017, when they look into sequence similarity and try to find some sort of motifs there. There is also this iSmart approach and many others in thinking a bit in line of the sequence similarity. But there are a lot of different sequences, and some of them share similarity, share the binding specificity. So it could not be all the sequence information alone information is necessary. Maybe if we look at the actual binding in the contact sites of between the receptor and the antigen it binds to, maybe it is actually a short stretches, short subsequences that are relevant and that are our kind of data representation for machine learning. One such approach was this paper by Osmeyer and colleagues, where they try to predict cancer status, where they look into 4 sequences from T-cell receptors. Alternatively, there are methods that look into physicochemical properties of these receptors, and sometimes those are also based on property of subsequence properties of subsequence of these types of receptors. So there is also, for example, a TCR match that looks into k-mer similarity and then averages the k-mer similarity across different k-mer lengths in order to match the sequences against the sequences with the known specificity. There is also other approaches that look into K-mer frequencies in order to predict this type of information. However, we can hypothesize that this might not be enough. Are the K-mers all there is? It could be that it's not. So in order to predict receptor specificity, some other approaches look into modeling the antibody-antigen interactions, and they try to model the relations between different antibodies and different antigens, and then try to kind of project that into unique or to come up with a unique representation that could be later used for predicting receptor specificity. There are also other approaches that look into how we can enhance the receptor specificity prediction, not just by using sequence information, but enriching this information with structural information as well. So, for example, this deep AIR approach, it includes the way to represent gene information, sequence information, but also structural information, as it was predicted by AlphaVol2. And there is also other approaches that look into predicting structure, and then based on this structure, try to do, for example, virtual screening of antibodies against a target antigen. Finally, this is some very specific and concrete things that we look into, and these are all manually set features based on what we believe is relevant for the sequences. But alternative is actually learn latent representation, or alternative to learn an alternative representation using sequence, and maybe include gene expression data as well, as it was done in MV-TCR that was recently hosted as a preprint. That is one option. Other approaches also look into what kind of information is necessary in order to be able to provide to learn this type of latent representation that could be used either to predict receptor specificity, to track the changes in the repertoire, or to cluster sequences, do maybe batch correction based on these latent representations. Finally, in order to predict the binding, it might be easier to not look into the only the sequence information, but instead use the structural information. But structural information is predicting the structure of antibodies is not an easy task. So what some research groups are doing is trying to predict the structure of antibodies, and then use that information to predict this structure in order to discover epitope or antigen specificity. So there are some different approaches to doing this. Some are based on graph neural networks. Some are based on other types of like convolutional networks. Some are based on language models. And some try to combine information from multiple different models, and also then, by doing that, they're able to estimate the error rates that is like where the different models disagree about the position. That information can also be provided to kind of estimate the uncertainty of the prediction made by the machine learning model. And as mentioned before, some of these previous models are based on language models in order for predicting the structure of receptors. And it's easy to just say this is something that works well for in machine learning in general, for natural language modeling. So this is something that we want to apply also in AIR setting. But what is important is to also kind of look into and formalize a sort of antibody language. Why do we want to do something like that? Is because we want to be able not to just model the structure or perform our task, but we also want to be able to get some sort of biological insight into the data, into this the structure of sequences, into the binding, and so on. So in order to do that, we could maybe benefit even more from language models by formalizing this antibody language with clear correspondence between the natural language task and properties in the, for example, antibody sequences that would allow us to construct, to learn what the model has learned, to be able to interpret it, and hopefully be able to design the therapeutics based on the obtained information. There are also some generative models that are based for adaptive immune receptors. Some of them model the VDJ recombination process, whereas some try to model antigen-specific antibodies directly, for example. In general, this modeling of different antibodies is something that we would typically want to do in order to enable design and streamline antibody design. And what we typically look into there is epitope specificity. So we want antibodies so that they're specific to the target of interest to provide the desired affinity, but also to look into the different parameters that describe the developability parameters, so how possible and how easy it is to actually generate these antibodies in experimental settings. There are many public repositories that provide repertoire sequence data, both the structural and the sequencing data itself, and also some. It is possible to generate synthetic data in order to look into how these models work and to hopefully streamline this part. There is another task that we might be interested in is predicting the antigen presentation on MHC complexes. So we want to predict peptide MHC binding, and we want also to predict binding of TCR to the peptide MHC complex. And there are many approaches based on deep learning for this task. For example, as was recently reviewed by Nielsen and colleagues, we might want to have different sequence information, some information that is coming from the side, some information that is coming from the MHC side, and then want to be able to combine this information to predict if the binding will occur or not. Finally, we might want to diagnose immune-related diseases using receptor repertoires. So instead of looking into one, we want to look at a collection of receptors in one individual and be able to diagnose different immune-related diseases. Repertoire classification as such is something called multiple instance learning problem as defined in machine learning. And in this case, we have a set of instances, a set of sequences, only some of which are specific for our disease. And the presence of these specific instances and maybe some other information determine whether the repertoire reflects if a person has a certain disease or not. And from the machine learning side, there was this nice review by Carbono and colleagues that describes multiple instance learning in a more formal setting. But there are also a lot of approaches for diagnostics that are mostly based on trying to explore this sequence, this structure that exists in the repertoire, this multiple instance structure, that try to use that to be able to better predict diseases. And as such, they typically perform much better than the methods that do not take the repertoire structure into account. So different machine learning methods have different underlying assumptions. Some of these assumptions are biological assumptions, what kind of information we need. Some are more subtle, more implicit, in that if we look at the k-mers of different length, what does that mean for our model, what our model can learn and discover and what it cannot do. So that is something that should be a conscious choice by the method developer and should try to reflect the problem domain as much as possible. So this is something that we want to be able to do, to have machine learning methods that reflect what the problem of interest and that are able to predict well on the data that we have. And with that, I'm returning the headphones to Maria.
So what should we do if we want to develop a machine learning method that work well to unseen receptors and repertoires? So here I show a very naive way to perform machine learning analysis for AR data. We can take an experimental data set that applies several machine learning approaches, estimate the accuracy of every method, and then just pick the one that works the best for this specific data set. But there are some potential problems to this type of analysis. First of all, the experimental data has its own specifics when we are talking about the method development. As we know, the dataset size for experimental data might be not that large, and for machine learning, we want to have as much data as possible. And also, we don't really know if the sample that we are working with is it actually representative. And then we cannot easily slightly change the experimental data to see how the method will work with slightly changed data set. It's called sensitivity estimation. And also, we don't have the ground truth information for experimental data, and we cannot easily say what the model actually learned from the data, and can we generalize it to a new unseen data set. When I say this generalizability problem, I mean something like this. Imagine you have a data set, maybe experimental data set, and you know that your machine learning method works perfectly well with your data set, but what if you try to apply the same method to a new set of receptors or a new individual, for example, for some clinical properties, for clinical things? One potential way to avoid these problems or accurately estimate the performance of our model on a different data set can be nested cross-validation. So nested cross-validation helps us to estimate the quality of our machine learning approach using the splitting of the data set into training, validation, and test. So we optimize our machine learning model on the training and validation set, and then we calculate the performance, like the final performance of our model, to a new test data set that was unseen before by our model. So that can make the accuracy estimation a bit more. But there are also other ways to verify that the machine learning model actually worked in our field. So as I mentioned before, we can do the training and dataset split to estimate prediction on the new unseen data. Then if we have some prior knowledge which is well studied, then we can use that knowledge and experimental data to compare what we learn, what are the hypotheses that we can generate with our new machine learning approach to what is already well known in our field. But if we don't have such well-studied information, then we can also try to simulate the synthetic data where we can implant all the knowledge of the field that we actually have, and then we can see what the machine learning model can generate from that data and what was in this synthetic data set, what was the knowledge in that synthetic data set. So what can be a prior knowledge in our AIR case? For example, it can be the VDJ recombination process. So we know that the receptors, they are generating during the VDJ recombination. And if we can somehow also include the binding specificity into this model and then generate a data set that takes into account all of this, then we can not only to estimate the accuracies of several machine learning approaches on one experimental data, but we can also see if the model actually learned what we encoded in the data as the binding, as a VDJ model, etc. So we aim to achieve both high accuracy and is the ground truth was actually learned. There are the most known tools that can simulate VDJ model. One of them is AIRR and Olga. The other one is ImmuneSim, so they both can do accurate VDJ recombination simulation. And Olga and AIRR can also estimate the generation probability of every receptor with respect to the model, but ImmuneSim, for example, can also implant a gapped chimer into receptor, so which can also correspond to the specificity of this receptor. We can use VDJ recombination and this implantation idea to profile AIR machine learning methods, for example, logistic regression on a range of basic data sets. So we can implant those short chimers with different frequency, and then we can see if those motifs were actually detected by our method. And with this study, we've shown what are the parameter boundaries for different chimers. But in real life, we think that immune signal can be much more complex. And from the previous studies, we know that an immune signal can be a gapped chimer, or in more general way, it can be a motifs or a distribution over nucleotides or amino acids. From Emerson study, we know that those specific receptors can be even the full-length receptors. And in the most general way, this immune signal, this hidden information, can be any function that takes a receptor and then returns true or false, whether the receptor contains immune signal or not. So here we formalize the immune signal definition for the AIR data. So we start with the motifs. A motif is a distribution over nucleotides or amino acids, but then for the immune signal, it's a motif plus some immune-specific information such as V gene or J gene or IMGT position of this specific motif, and then we can combine this set of immune signals with the disease label later. Of course, if we want to make a simulator that can do all these signal simulations, we should take into account many different aspects. So ideally, our synthetic data should have similar biological properties as experimental data. For example, the synthetic data should have similar distributions of some biological statistics, such as generation probabilities or VDJ distribution, gene distribution. Also, we don't want to break the biological properties of our data. For example, we know that the immune receptors have conserved area in the beginning and in the end of the CDR3 region, and we don't want to break that. Also, we want to simulate immune signal of any complexity, and our ideal simulator should do that. And one more thing that complicates the simulation is that these different immune signals can be receptor can contain several immune signals, and also those signals can be overlapping within one repertoire. So it's not very easy to make a perfect simulator that will take into account all these things. But currently we are working on such a simulator. It will do this signal implantation on the receptor and on the repertoire level. But also if you want to work with some structure, you can use absolute. You can generate synthetic three-dimensional antibody antigen structures using absolute. Then you can introduce all the possible signals that you want your machine learning model to detect later. And then that will be your ground truth for the machine learning method that it should detect. And then you should check for the high accuracy, both for the high accuracy and what was actually learned by the model. I think that this synthetic data approach can be a really nice sanity check for our model, because if our model cannot detect what we think it should detect in an experimental data, and something that probably it will not also work with the experimental data. So ideally, we think that AIR machine learning approaches should be developed in parallel on the experimental and on the synthetic data. So, Milena?
Okay, so sorry about the technical difficulties. They were not there half an hour ago. We talk about this simulation stuff and talk a lot about how we can compare different machine learning methods. But in order to do any of those things, we first need to be able to kind of reproduce the existing studies. And there are many different studies, and they differ in the languages they were implemented in. They differ in whether the code is available or not, and is it just the description of the algorithm, or is it actually the open source code? So we have this different amount of information for these studies, and we want to make sure that not only that they work as described, but how they perform on our own data sets or new data sets. So last year, there was this DOME paper that included a set of recommendations for reproducible supervised machine learning in biology, that looked into different types of information that are kind of suggested to be provided by different method developers, so that these methods can be both reproducible and properly reported. And this includes the recommendation related to how we provide the information in the data sets that were used, what was used for training, what was used for testing. Then it includes how we do evaluation, how we do optimization in order to avoid fitting our method too much on the current data set that will not then generalize to new data. It requires of us to include the description and the choice of the model, possibly release the source code and other information, and then to be very specific about how we do evaluation of different methods. And for example, the cross-validation is one of the good practices in that regard. We also looked a bit into this from our side in our group, and one thing that we developed is this platform called immunoml for development and transparent evaluation of different AIR machine learning methods. immunoml is open source. It's publicly available. It's available on GitHub. It's available as a Python package. And what it does is it formalizes some machine learning workflows that are relevant in the AIR setting. So it provides the workflow to train a machine learning model that is based on the AIR dataset and the detailed specification files to describe everything that has been done in a great level of detail. We can do a bit of a simulation, but that is just a subset of that. Then we can apply the trained models, and as output from the analysis using immunoml, users get HTML reports, trained methods, or synthetic datasets that were provided. So this is one attempt at kind of helping the community by developing this infrastructure around their methodology development, and I hope it can be useful. So with this, we kind of want to be able to ensure that to implement the procedures that allow us to estimate how well our methods perform on the new datasets, or on the datasets that we have in a proper manner, correct manner, to test it to account for overfitting, underfitting, and similar issues that can happen in machine learning. But the question is, if we actually, instead of that, if we actually develop a new method and we are happy with it, what is kind of the next step? So in terms of developing a diagnostics, what we would want typically is to, we start with some sort of source population that has healthy individuals and diseased individuals, and they have some properties, some sort of distribution over their characteristics, like age distribution, how many are diseased versus how many are healthy, and so on. And then from that population, what we typically do is we select a set of participants for our study, and this is our study cohort. Then we say, okay, now we do sample collection, we do sequencing, and we obtain a certain set of repertoires that is, again, partially reflecting what is happening in the individual's body. And I say, okay, then using this information, we want to be able to build a machine learning model, and we can estimate its performance, and we are quite happy with what we did. So now, if that works, what we typically want to do afterwards is actually use that on a new kind of some sort of new population. On it could be now we want to apply it in the clinic, or it could be just we want to use it for a different data set coming from a different group. And what is problematic with this is that if we look into the different, as called here, variables of interest, but it could be age, HLA, or other information, they will typically differ between populations. And or it could just change between different populations. So how, what do we do with machine learning in this case? Because machine learning is defined to, as we discussed it before, it was defined to work on data that is independent from each other and that has the same distribution on for the data set that we trained the model on and where we want to apply it. And that's not something that necessarily happens when we come to the application settings. So there is something called the causal inference framework. And the causal inference framework is more concerned with formally describing the data generating process, like our biological mechanism, in order to discover some sort of causal effects between different variables in this process. So is that if we have one variable, we want to estimate how it causally affects another. So this is just how this other variable changes how the changes in the first variable affect the other one, provided that everything else is the same. And we talked about how machine learning estimates statistical associations, not causality. And causality as such doesn't matter too much for prediction tasks, but it's still when we look into new data that we obtain, or when we apply to new populations, if we look into causality, it can help us formalize and describe these changes, and maybe solve some challenges even in predictive setting. And if we look into an example of a causal model describing like what is that's happening in biology for a viral infection, we can draw like different variables that represent the different concepts of interest, like age, genetics, HLA, the repertoire. We can see that there are different relations between these variables. So what can happen is there can exist some sort of a selection bias in our data, which can happen anytime that we have some sort of preferential selection of study participants. It can introduce spurious correlations in our data, which is something that we don't want to happen. Then we can have confounding bias. So those are the variables that influence both the repertoire and the immune state, for example, that we want to predict. Or we can have different batch effects, timings of measurements that influence there. And causality kind of defines a bit how we can, what do we need to control for it, want this to be stable or not. So this is just something thinking in line. How can we guard against the changes in the data, different data sets? So with that, I think we could wrap it up. So just some points that I hope came across clear from our talk, is that there are many machine learning methods that are applied to AIR data, but it's more complex than just choosing an off-the-shelf machine learning and putting it on the AIR data, because AIR data has complex biological structures, both in and in terms of repertoires. There is a large the repertoires, the receptors are both very diverse, and we only observe a small portion of the potential receptor space. And there are other variables that also influence what is happening in this case that could complicate our machine learning methods. So one thing that we hope to show was that it is necessary to benchmark these developed methodologies on both synthetic data and on experimental data, so that we can do a bit of sanity checks in the synthetic data case, and then see how it actually performs on the experimental settings. And in order to be able to develop methods that will actually work in and be possible to apply in clinical settings, for example, we need large-scale experimental data, and we need to know the ground truth so that we can validate our algorithms. And with that, we would like to thank the other people from our lab that kind of work a lot on this topic. And yeah, thank you for having us here and giving us the opportunity to present today. I think we could try to...
I will switch off the mic.
Yeah.
All right, well, I'm back anyway, and I hope our speakers will soon be ready. Are you ready to answer questions? Looks like you're both muted.
Okay, can you hear me?
Yeah, we have one of you. Well, maybe two of you because we're getting echo. But
A bit of setup issues.
well, you're winging it very well. We can hardly
Should we
tell
try
there's
to
a technical issue. All right, are you
answer
ready?
the question?
Yeah, are you ready to answer questions? I will start with the first one.
Okay.
Do you think there is
you want
enough
to start?
information encoded in the receptor sequence?
Yes. Should we start from the first question?
Can you hear me?
Do you think there is enough information encoded in the receptor sequence alone for ML to be able to predict antigen specificity? Or will this always require some structural information? I mean,
that's a good question. And I think the fact that we can predict structure from the sequence information might mean that this could be something that can be used also for antigen specificity prediction. So what some of the groups are doing is to actually develop a structure prediction method and then use the predicted structure to predict the antigen specificity, which could be one way to go around it. I hope that answers the...
Should we also cite ABDB there?
Yes. So, yeah.
Okay, I'll go ahead and ask the question.
Are these immunoseq available in AIR? Do you mean the data that is available or the methods that are available? So I think there are quite many public data sets that you can use. And also some of the simulation tools are already available. So for example, AIRR, Olga, ImmuneSim, they are already published. Absolute, you can find it as a print, but it will be soon available as a publication. The simulation approach that I was talking during the presentation, we hope that we will also have a preprint soon. So if you can specify what you mean by immunoseq, maybe I will reply in more details.
Yeah. So I think we can just move on.
Yeah.
So for the diagnostic scheme, are public immune receptors more useful than unique receptors? Some approaches look into only public receptors. For example, in the Emerson, in the paper by Emerson and colleagues, they look to discover sequences that are associated with disease based on looking only into public sequences, and then that appear that are enriched across repertoires. That is definitely one possibility. However, a very small percentage of sequences are public, and but not a small percentage of sequence are necessarily not all of the antigen-specific sequences need to be public. So there is probably some other information that could be used also from unique receptors that are shared across the unique receptors that share the antigen specificity. So from some analysis of structural information, it was shown that some of the sequences, that the actual binding happens on a subsequence level, and maybe it is enough for those to be shared across receptors. Maybe it is a bit more information, but it's probably something more detailed than actual full sequence, full unique sequences.
Can I add just a bit? So in my opinion, they both matter, the public and the unique one that are specific for disease, but maybe they are matter in a slightly different way. So I think we should take into account both. So we cannot just say, let's zoom into the public receptors. Those are the most important. I think all of them are important.
Okay.
Considering therapeutic applications, do we know the immunogenicity of them? Do we mean the specificity by immunogenicity? No. Well, there are quite many different data sets and databases. For example, there is this VDJDP MCPass database for where we have the information about different receptors and then the specificity to the antigen. And sometimes we also have the information, the sequence of the antigen. So it is collected from many different papers, from many different studies in one database, so we can use that. But most of the time, I would say for the therapeutic application, we just have the AIRR-C data. So we have just a FASTA file with bunch of receptors, with bunch of strings, and we don't really know what is the specificity of each. In future, we hope that using the Libra-seq and other methods, we will know the specificity of every receptor, but for now, we only have the sequences.
Okay, so next question is to what extent are models that use structural information for the prediction of receptor specificity reliable? The structure is predicted and not experimentally proven, so its accuracy will always depend on the accuracy of AlphaFold or any algorithm that was used. So yes, of course, that is a challenge in this regard. In terms of predicting the receptor specificity, that information might be experimentally validated. That can be kind of one way we assure that our structure-based prediction of specificity kind of works. Is it a solved problem? Probably no. Can we predict it decently? Probably yes. But I think we need kind of more better ways to explain what is happening there and to actually validate all these predictions that we have gotten so far. So I think this is definitely going in that direction, but probably still some external validation would be needed.
Since there are so many models and ways to approach sequence embedding, how to know which one to choose? That was a really nice question. I mean, probably use some synthetic data, because also different methods and different encodings, embeddings, they use different assumptions about your data. For example, if you use k-mer encoding, if you just cut your sequence into k-mers, then probably those encoding will not be powerful enough to detect some information that cannot be easily translated into those k-mers. So
yeah, I think there are many models, there are many ways. What is needed are like benchmarking efforts in order to kind of consolidate this and be able to say something more definitive. But for that, we also, as I said, need the ground truth data that is like to confirm that even if the model performs best, that it is actually learning what it's supposed to be learning. So this is an open and ongoing research question.
Yeah, also, I think it will be really nice if the AIR community will benchmark all the AIR machine learning algorithm on a unified set of data sets with different properties, and both experimental and synthetic, but this is not done yet.
Yes. Yeah, and this is the follow-up question. So the way to check which model is the right one is to run as many embeddings as possible. Maybe not as many as possible, but I think it is kind of necessary to do extensive benchmarking, because most of the methods compare against very few other methods. And another component that we have to look at is what we believe about the domain to be true, because off-the-shelf stuff doesn't necessarily work well. So what we want is to see what methodology that we have best reflects our domain knowledge, and then compare those into like on actual either experimental or synthetic data sets. Okay, so there was a comment that a newer version of AVDB data is coming up soon. That is very good to know. So one more question is, how did you approach the design of immunoml so that the majority of AIR algorithms can be used? So what we try to do is to define what needs to get, what is the input and what is the output of different methods, while leaving the actual method implementation completely up to the method developer. So what we provide is, if someone, we try to also implement some methods inside immunoml that were published before immunoml was there. But we also hope that the community would use immunoml. So also for development of methods. And the workflow of someone using it for development would be that it is just necessary to implement your own method the same way you would elsewhere. But immunoml would take care of representing the data. It would take care of importing the data in appropriate AIR format and things like that. Doing this cross-validation stuff, reporting the details, and all those things. So on the website, we also have developer documentation where we describe this in great detail and are very open to community feedback and discussions.
I think that's all.
Yeah, those are all the comments. There is one more. Wouldn't benchmarking fall into the same issue as applying models in different populations? Different subset of sequences from different samples may yield different results for benchmarking. Well, or do you want to answer that? Or
yeah, maybe you can start, and I will add a few words from my side.
Yes. So I think it depends how you define the benchmarking. So what you could do is not compare the exact trained models, but instead compare the algorithms, train them all on the same data, and compare them all on the same test data. You can do that on one data set, on multiple different data sets across certain, like increasing, for example, level of complexity, or differing with the different assumptions of how the data, the signal in the data actually looks like. And it could also be how well, so this is like something we call internal validity, right? Like how well your method performs on this data that looks the same. And then on top of that, you can also look into what happens when you change the distribution of your data, and when does the method stop working, does it always work? What are those kind of scenarios? So you can define benchmarking in both of those.
Yeah, so no one ultimate algorithm that will work perfectly well on different types of data. Probably one method will work when we have some confounders. The other method will work better if the immune signal or something is defined as a k-mer. The other method will work better if it's defined as the full-length receptor, etc. So yeah, it's like saying that there is one statistical test that will, you know, work perfectly well with all the possible alternative hypotheses. It's never like that. So I think it's something similar to that problem.
Yes. Thank you for interesting questions.
Yeah.
All right. Well, I
think we've been
stop because they can't hear me.
We cannot hear you, probably.
Yeah, they can't hear me because they don't have the headphones.
Probably we need to stop.
Yes, so I think you need to stop. But I do thank our speakers for providing their insights on machine learning for the analysis of adaptive immune receptors and repertoires. And I thank you also for joining the webinar today. An on-demand version will be available in a few days. I will send a link by email to everyone who registered. So you will be able to find this in a day or so. Please feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.